it into two hosts. The snail, which is an intermediate host, and the human. Okay, so as I mentioned, there are two hosts. The snail, which is intermediate, and the humans, which is definitive. And for our PowerPoint presentation, we will concentrate on the activity of the immunobiological relationship of Schistosoma mansoni and the humans. Because both represent various amounts of literature, but the literature regarding human is more complete and it's more adept to our benefit. So, go ahead.
Okay, to start with, we're going to give you a general overview of our report. So first, Schistosoma mansoni is also known as commonly known as blood flu, is a parasitic trematode which belongs to the family Schistosomatidae. The causative agents of the disease is schistosomiasis. Uh, Schistosomiasis is a disease. Schistosomiasis is a disease which is notorious for not only neutralizing strongly mounted immune defenses, but also this S. mansoni is famous for using the immune response of the host to survive and proliferate within the body. So, Schistosoma mansoni together with Schistosoma hematobium and Schistosoma japonicum is responsible for putting an estimated number of 600 million people spread over 74 countries, which 280 people die per year in sub-Saharan Africa alone. That's why this disease is medically significant, and a lot of research should be put to study it. As for the epidemiology of Schistosoma mansoni, usually schistosoma, the ones affected are those that belong in the low economic or in low economic and educated places. So since Schistosoma mansoni is transported in fecal matter infected by the parasite, usually the Schistosoma mansoni is spread geographically within water reservoirs or bodies of water infected by this fecal matter, contaminated by this fecal matter infected by Schistosoma mansoni. So, for example, the areas usually affected by Schistosoma mansoni are those near bodies of water such as rivers, contaminated rivers, or let's say urban rivers, to be more precise, and those that live in low educated and economic areas such as water areas. As for the anatomy, Schistosoma mansoni is dioecious, meaning there are male and female species in different individuals. So, and in Schistosoma mansoni, since it's dioecious, sexual dimorphism can be observed. So, for males, the males appear to be short and stout with a longitudinal canal found at the ventral side called the genicoforal canal, in which the long and slender females reside. So in males, usually there are only 6 to 9 testes per organism, and and the females usually contain a few elliptically shaped eggs with lateral spines. And these anatomical features, the male and female, are species specific. So what I mean is, it's only that feature is only specific to Schistosoma mansoni. Like for example, in Schistosoma japonicum, um, the the males contain only 1 to 2 testes. And also for the shape of the female is specific to Schistosoma mansoni, since the ovary is located halfway anterior to the body. So and with the ventillaria posteriorly located to the body. Okay, so this is the hemicophoral canal from which the female resides. And as you can see, the males are short and stout, while the females are long and slender. And this sexual conformation, this sexual conformation is observed because for sexual reproduction and movement within the vasculature of the host.
I am now here to discuss the general life cycle of the Schistosoma mansoni from its transmission to infection of the intermediate and the human host. So here is a picture depicting the life cycle of the Schistosoma mansoni from wherein you see a free swimming Cercaria entering the human body. But let us not dwell on that first. So, how is Schistosoma mansoni transmitted? Schistosoma mansoni starts its transmission once the egg, contained in fecal matter, disposed in bodies of water, is introduced in bodies of water. In optimal conditions, the egg breaks and meracidia emerges from these eggs. The meracidia then penetrate and reside within the intermediate host, which is usually the snail, specifically the Biomphalarian snail species. In the Biomphalarian snail species, asexual reproduction commences, forming two sporocytes, which is the daughter and the mother sporocyst. After asexual reproduction, the sporocysts develop into Cercariae, which is the free swimming stage, of the parasite, wherein because they are free swimming, they could directly penetrate the human host species. Now, upon penetration, the parasite travels within the body, and once again, the cycle is repeated when eggs are released again to bodies of water. So, now we shall discuss specialization and specificity of host-parasite relationships. Specificity and recognition of the host depends on factors which are the spontaneous behavioral patterns and behavioral patterns in response to signals elicited by the host itself. To give you a clear overview, the signals transmitted by the host, for example, the human host, cannot travel very far chemically nor physically. However, the morphological and behavioral characteristics of Cercariae still allow it to reach the host, its desired host. So, with the help of other factors listed here, such as environmental cues including temperature, gravity, water current, light, and time, the Cercariae is able to reach the host. Now, one peculiar trait of the parasite is that it relies on time. How is this exhibited? Now, Cercariae could actually choose when to what species it would like to invade. For example, Cercariae is dependent on time in these situations. If the Cercariae wants to penetrate a mouse species, the parasite usually goes out into the area or the body of water at 4 to 5 p.m. And if it wants to infect a human host species, it usually is observed from 11 to 12 at noon. Now, when the parasite already reaches the host, recognition and attachment is dependent on two stimulus, L-arginine and warmth. Now, how does this happen? For warmth, if the Cercariae is able to detect a change in even the slightest 1 degree Celsius, an increase in 1 degree Celsius, the Cercariae identifies it as an optimal environment to invade. L-arginine, on the other hand, helps in directing the movement of the parasite towards the host. Because as we know, the Schistosoma manzoni invades the blood vessels. Once the parasite is able to recognize in a natural host, it is able to keep, it is able to penetrate the host with the help of another lipid, namely the ceramide, which is found in the epidermal layer of the skin. So ceramide helps in ensuring the contact or makes the attachment longer. Next, so here we see the layers of the skin. Once a parasite enters, it starts from the stratum corneum, and then it is directed to the stratum granulosum, and eventually to the stratum basale. So, what does this mean? As you see, there is a yellow arrow. This is L-arginine. It says that we are able to find out that the Cercariae directs its path towards areas with high L-arginine content. So, as we're reaching the stratum basale, it makes its way to the dermis. But before entering the blood vessel, we must note, however, that the forked tail, as you can see here, is lost by the parasite before entering the blood vessel. Now, once in the blood vessel, the parasite is attracted to still L-arginine and to a lesser extent D-glucose. L-arginine is found in blood serum, and D-glucose is found in the endothelial lining of the blood vessels as well as in proteins such as bradykinin and fibronectin.
Okay. So the significance of Veron's part is to explain how Schistosoma manzoni recognizes its host. Remember, with the main definitive hosts of Schistosoma manzoni are mammals, and they are warm-blooded, which is why their temperature contrasts greatly with the surroundings. So the warmth of the host is a signal for Schistosoma manzoni to attach to them. And for L-arginine, from the outer layer of the skin towards the dermis, there is a gradient of L-arginine, and without that, Schistosoma manzoni would not know where to go. So it points its body ventrally to the concentration of L-arginine and moves towards there. And again, in the blood vessels, in the different L-arginine containing proteins of the blood, without it, the Schistosoma manzoni again would not know that the blood vessels is over there, because in the endothelial, the inner lining of the blood vessels, again, are different proteins such as bradykinin, which contain L-arginine. So my part involves the immune activity of Schistosoma manzoni. It can be divided into the migratory phase, which refers to the penetration of the Schistosoma manzoni towards the human host, the acute phase, which is marked by the beginning of egg production, of sexual production, and the chronic phase, which may last up to 40 years because the Schistosoma manzoni resides in the mesenteric vasculature, and within it controls the different immune defenses to its advantage. So next. So here's a graphical summary. So the important part here is that the Schistosoma manzoni induces two types of responses, the Th1 response and the Th2 response. Both are immune responses that produce different types of interleukin with different effects. So it is important for Schistosoma manzoni to become successful, but it is important that they balance the amount of Th1 response and Th2 response. If there is too much Th1 response, the host will die. If there is too much Th2 response, the host will die again. So the migratory phase, as mentioned by Veron, is the part where the Schistosoma manzoni goes from the epidermis to the dermis. And again, it's still the schistosomal part when it just shed its skin. Therefore, it is still not yet sexually mature. And during this phase, the body of the Schistosoma manzoni, next slide. The body of the Schistosoma manzoni, next slide. Okay, again, the Th1 response will induce a different set of cytokines compared to Th2, which I will specify later. So next. So the first one is the Th1 response during the migratory phase. The schistosoma mansoni itself attracts macrophages, which produces tumor necrosis factor. And this TNF will induce other leukocytes to produce other interleukins. The main interleukins of the Th1 response are the IL-1, IL-6, IL-12. The common factor between these cytokines are they are inflammatory. They are pro-inflammatory. That's why if you have too much Th1, the host will die because of excessive inflammation. So next. So next, the acute phase. Let's say the Schistosoma mansoni has matured and begins to produce eggs. The eggs induce a Th2 antigen response. And part of this Th2 antigen response is the interleukin 10. This interleukin 10 serves to control the activity of Th1. This is why during the migratory phase, Th1 increases because only the Schistosoma mansoni adult is present. However, after sexual maturity and the eggs begin to be produced, the interleukin 10 produced by the Th2 response modulates this Th1 response, and hence the activity of Th1 response onward is much less than compared to the migratory phase. However, next slide. Okay, so here we see that egg antigens secrete sugars and proteins that are detected by dendritic cells, and these dendritic cells activate IL-10, which is what controls the Th1 response. So without the IL-10 produced during the Th2 response, Schistosoma mansoni would die. Because as I mentioned, without the balance of Th1 and Th2, the host will die for different reasons. For example, again, for Th1, it is pro-inflammatory, which will cause excessive fever and other symptoms related to inflammation. For example, back one. Again, no. So again, this is why the acute phase, when the Th1 is still high, it's called Katayama fever. Because the IL-1, IL-6, IL-12 are pro-inflammatory, and these, specifically IL-6 in humans, are febrile. They cause fevers, hence the name Katayama fever. So this shows the transition between the migratory phase and the acute phase. So in the beginning of the acute phase, there is Katayama fever. However, as the response continues, see, Th2 is going too high. But after this, it goes down. This part onwards is the chronic phase. So chronic phase. Okay, so it's the chronic phase. So if you have produced too much Th2, what happens is eggs that are secreted by the eggs secreted by the adult Schistosoma mansoni travel the blood circulation, and they end up in the liver. They are stuck there. And they are bombarded by Th2 responses, which involves T cells, B cells, and other types of leukocytes. And this, specifically, they produce interleukin 13. Interleukin 13 causes granuloma formation. And if you have too much granuloma formation in the liver, it will cause several diseases in severe cases of chronic disease, such as portal hypertension in the liver, ascites, as seen in the pictures before. So again, if you have too much Th2, the host will die because of the aforementioned reasons. This is why, again, for unknown reasons, interleukin 10, this shows the activity of interleukin 13 and interleukin 4 in inducing macrophage response to produce protein, which is an amino acid integral in the formation of collagen, the main protein in granulomas. So, next. So here's a picture of the granuloma, and the granuloma composed of the different parts, the collagen and the other leukocytes. So as I was saying before, it involves a balance of Th1 and Th2. If you produce too much Th2, the host will die because of too much granuloma formation. However, as you remember in the graph, diba tumaas yung Th1, pero may konti pa rin siyang Th1 response. Different pro-inflammatory factors such as the IL-1, IL-6, as those I mentioned before, serve to counteract interleukin 13. As shown in studies, application of these interleukins would prevent the formation of granulomas. Next. Before. So, no, side. So again, however, the activity of these anti-IL-13 interleukins are still theoretical. Even though this is the case, the production of granulomas is still important. Let's say you have an egg, just an egg. It has antigen, it produces hepatotoxins. But if you have granulomas, these granulomas will sequester these hepatotoxins, thus aiding the survival of the host. So the antigen produced by the Schistosoma mansoni also induces antibodies that will neutralize these hepatotoxins. So here again, we see another instance of Schistosoma mansoni using the immune response to its advantage. So for other examples, not necessarily related to the Th1, Th2 response, is 2.
So, Jarvie talked about how Schistosoma mansoni balance the immune system for their benefit. But now I'm here to talk about how Schistosoma mansoni directly sequesters or uses the immune system for its direct survival. So the first is that you may have heard it from what Jarvie mentioned earlier, that they stay in the adults live in the mesenteric vasculature. And because it is an aerobic environment, it is also, the Schistosoma mansoni are prone to oxidative damage. Now, in a study, it was found that Schistosoma mansoni survive oxidative damage because they have they produce a superoxide dismutase called TGR, or thioredoxin glutathione reductase, which is very similar to the human homologs, thyrodoxin reductase and glutathione reductase. So we see that here that they use similar modes for survival in humans. The next would be in another study. It was shown that Schistosoma mansoni are able to secrete neuropeptides, which are immunoactive. First, they produce ACTH, adrenocorticotropic hormone, and beta endorphin. And then ACTH is naturally cleaved by naturally occurring human neutral endopeptidase, which can become alpha NSH, or melanocyte stimulating hormone. Now, ACTH has been known to inhibit production of interferon by T lymphocytes and activation of macrophages by interferons. And when I mentioned about beta endorphin, they could not clearly identify what it was doing in the immune system, but rather they assumed that it could be for signaling purposes. So the next would be alpha melanocyte stimulating hormone, which is produced. So it is able to prevent adherence and locomotory activity of polymononuclear cells and monocytes, which are important in the complement pathway. So this is an example of how Schistosoma mansoni can survive the immune response of complementary pathway, of the complementary pathway. The next would be how Schistosoma mansoni sequesters the cell, the red blood cell, the erythrocyte, to survive other immune responses other than the complementary, such as the complementary pathway. So in this study, they wanted to know if resistant variations of Schistosoma mansoni were using masking techniques for their epitopes, meaning that antigens could not bind to their surfaces, or were they using something else? So they did this by first using a known substance, immunoglobulin G, known to have anti-schistosoma properties, antigens, So they used this, and then they found out that they still bound to Schistosoma mansoni adults. So this proves that they were not using a masking technique. Next, they noticed that there were similar cytosolic components from the erythrocyte found in a complex with Schistosoma mansoni. So what they did is that they lysed the erythrocyte, and then they separated it into two, the plasma membrane and then the hemoglobin rich factor, and then they cultured it in separate areas. They found out that it was only the membrane which provided protection against the immune system with a very high survival rate. So with further identification studies, they figured that there were two components. One was found in the plasma membrane. One was the DAF, or decay accelerating factor, and C3b CR1. So it was known that if you add trypsin, it would completely inhibit C3b CR1 pathway without interfering with the DAF pathway. But if you added pronase, it would do the same to the DAF pathway, not interfere with the C3b pathway. So when they did this, they found out that they proved that decay accelerating factor was being used by Schistosoma mansoni to not directly inhibit the complement system, but to protect itself from the cytotoxic activity, which was the product of the complement system. Since that is because DAF and the C3b pathways were both already discovered to inhibit the cytotoxic activity of the complement system. And then lastly, it was found out that we noticed that it was mentioned that Schistosoma mansoni use developmental cues cues outside the body and inside the body. These are the developmental cues. So an experiment with SID mouse and nude mouse, both of which do not have T cells, found that there was a decrease in fecundity for Schistosoma mansoni, or even inhibition of maturity. So this proves that the immune system is not only a hindrance to the Schistosoma mansoni, it's actually something that is very needed in its maturity and life cycle.
Thank you.
This is very interesting, and I do hope all members of the class listen to the group. Very interesting, well described, well explained. I really love the figures that you